So, good morning, everyone. First of all, I would like to say many thanks for this wonderful opportunity to share these challenging yet a little fascinating insights on epigenetics with you. At this point, I would especially like to thank Ulla Hasche, who has put in tremendous amount of work to make this conference happen. Thanks also to the entire organizing team. You have done an excellent job. I really appreciate your kind of help and support. As a starter, I would like to ask you to read the following conclusion. And think about it a few seconds. That gives a basis for us.
Just.
Okay. The microphone is upside down.
Yes. Wait.
Okay. Is it better now?
Yes.
Okay. Excellent. Thank you.
Like that.
Thank you. So, let's continue. One of the most well-known claims of evolution is, that's it. We are too familiar with this guy, with this picture, and with those Darwin's finches we are too familiar with. But in the next hour, we will learn that Mr. Darwin was seriously wrong. But we know that according to Darwin, evolution, natural selection work as follows. Some finches develop strong and thick beaks as a result of random mutations. The random mutation theory was devised by Hugo de Vries, leading them to become seed eaters. Finches with slender, means weaker, thinner beaks, become insect eaters. In this way, natural selection directs birds with different beak types to acquire more food, giving them a competitive advantage. They produce more offspring than birds that obtain less food, thereby reinforcing the trait in subsequent generations. This results in differentiation, which leads to speciation. But after the next hour, we will learn that Darwin was seriously wrong. However, this misinformation is taught in schools all around the world. I know that especially because I'm a teacher. Now let's take a look at what recent research says about the causes of changes in the size and shape of Darwin's finches' beaks. This article is from 2017, and I have picked up the most important observations and conclusions made by the researchers, printed with a yellow color. Distinct diets which could explain the differences in methylation patterns, as diet is known to influence epigenetics. And Darwin's finches, they are genetically pretty similar. And they also noted that DNA methylation can be a source of heritable phenotypic variation and more. Environmental stressors, such as pollutants, diet, pheromones, etc., can change methylation patterns. At that time, this was a novel new mechanism for them, and the field of epigenetics was just getting started. But here they list the three main forms of epigenetic regulation, which are biochemical tag, meaning methylation on DNA basis, especially cytosine base, and second, biochemical tag to the histone proteins, and third, small RNA molecules. They also observed that none of those mutations, now they are talking about DNA mutations, none of them were consistent with those changes observed with those changes within beak sizes and shape. No fixed differences in gene copy numbers either were observed. Today we know that diet is the most significant reason behind the changes in finches' beaks. So, if a finch starts to eat seeds, then its offspring will have stronger beaks. And if it begins to eat insects, then after a few generations, the offspring will have thinner and weaker beaks. We know that these rapid changes are not based on random mutations or selection. Excuse me, Ari, could you bring me a glass of water? Thank you. I think this example of so-called evolution is also familiar for most of us. The blind cave fish. It's been also used as evidence for evolution for decades. Is this based on random mutation and selection? No. Today we know that these changes occur rapidly, only in a couple of generations. The eyes are restored after the fish return to a light environment. And it doesn't take millions of years, only a couple of generations. It's based on epigenetic regulation and specific opsins, light-sensitive receptors that transmit signals between the environment and the cells of the fish. Third example, I think we are also familiar with this one, Italian wall lizards. Evolution believers often claim that we have seen evolution before our eyes. Even a new structure has been discovered. Now let's have a closer look at this interesting experiment. Researchers transferred 5 pairs of Italian wall lizards from Podkopiste Island to Portmercaro Island in the southern Adriatic Sea in 1971. Thank you. Their plan was to return back to the island after a few years, but due to Croatian wars, they were able to return to the island only after the end of the war in 2004. And when they came back, they were almost shocked to discover how quickly the lizards had undergone radical changes. Their behavior had changed, the jaws had lengthened, and most surprisingly, their intestines had developed a ring-shaped muscle called the caecal valve. All of this had happened in just a few decades. The reason was clear. The lizards had to switch their diet from insects to plants. The science didn't find any consistent changes in the lizard's DNA. And today we know that a diet influenced the expression of the genes and epigenetic regulation in the lizard cells. And what is the most important thing is that all the information needed for that adaptation was already present in the lizard cells. And the changed diet activated certain genes and deactivated others. They worked like switches. Here's a picture of the caecal valve, ring-shaped muscle in the intestines of the lizards. Okay, more examples. Did you know that the bee larva grows into a queen because it has been fed royal jelly? No changes in DNA, but the diet makes the changes through epigenetic regulation. The DNA of the workers and the queen is identical. The ant grows into a soldier ant due to an epigenetic signal given by the queen. No changes in DNA. DNA is identical between these two ants. More examples. The food that stick insects eat affects whether or not they grow wings. Again, no changes in DNA. We can observe change, but this is not evolution. This is something dynamic and reversible. Mammoths, the Colombian mammoth and woolly mammoth, their DNA is identical, but we have two entirely different mammoth so-called species, hairy one and the pale one. Nautiloids. Researchers have named these nautiloids as different species, even their DNA is identical. And the butterfly metamorphosis, 4 completely different looks, but the DNA is the same in all stages. So we should understand that DNA does not dictate the appearance, meaning phenotype, of an organism. And this one is very interesting. DNA does not dictate skin color. This was proved in 2016 when these 2 pretty girls were analyzed by their DNA. This was quite stunning discovery. DNA was identical because these twins are monozygotic twins. Again, we should ask what kind of information determines the skin color. The skin color is not written in DNA. These delicious cabbage-related vegetables are pretty different, but their DNA is identical. And these finches look very different, but again, their DNA is the same. The most important question for a middle school or high school biology teacher or any teacher who teaches biology. And here's the question. The human body contains about 300 different cell types. For example, skin cells, liver cells, muscle cells, and so on. And only a couple of exceptions, all of our cells contain exactly the same DNA sequences, meaning their genome is identical. The DNA is identical in our cells. So the question is, why does a skin cell have a different identity and function and task and job than a muscle cell, even though their DNA is exactly the same? And we understand that DNA does not determine the cell's function, and we should ask, what then does? We know that there are more than, or about 300 different cell types in our bodies, and virtually all cells have identical DNA. There are only a couple of exceptions, neurons and T cells. And we should ask the same question, why are cells different even though their DNA is the same? And now we are coming to epigenetics. Epi is a prefix. It's Greek, from Greek language, and it means on top of or outside something. That means information that is outside of DNA. And most of all, it means some kind of chemical markers, chemical layers of information. For example, the most known one, CH3 hydrocarbon group, means a methyl group, and we are talking about methylation. And they direct the reading, meaning transcription of DNA. They regulate several cellular mechanisms, and they can silence DNA and prevent it from being transcribed. Did you know that more than 95% of plants' genomes are silenced by epigenetic mechanisms? The information is already there, but it's just waiting for the adaptational needs. And without epigenetic markers, a cell is an undifferentiated stem cell. It's like a dummy cell. It can't do anything. It's waiting instructions, but it has no job. It has no identity. It doesn't do what to do because it has no program. So we know that a cell requires epigenetic programming in order to perform its designated function. And we also know that methylation stabilizes the genome, meaning that if there are some kind of aberrant methylation profiles, then we will face problems with cells. And as those researchers listed the three main types of epigenetic information, I can repeat them: DNA methylation, histone epigenetic markers, meaning modifications, and RNA signaling molecules and non-coding RNAs. DNA methylation is the most studied form of epigenetic information. Even a single methyl group might affect DNA transcription and further how a protein or RNA molecule is produced. There are specific regions in the genome with a high concentration of GC pairs side by side. There might be hundreds or even thousands of them. These regions are called CPG islands. They can also contain many methyl groups and play a very important role in so-called gene regulation. For cell differentiation to occur in a controlled manner, methyl groups must be precisely located to ensure that the cell's mechanisms can read DNA from the correct locations. The cell, therefore, requires epigenetic programming to function in its designated role. You may have learned that there are so-called genes in the genome. Sounds familiar. And that one gene produces one RNA, which produces one protein. Now it's time to forget this old dogma. I mean, really, forget it. It's outdated. A cell can produce thousands of different proteins from the same DNA sequence without changing DNA. For example, human cells have less than 20,000 DNA sequences read for protein production, but the number of different proteins in the human body is up to millions. This incredible information handling mechanism is based on so-called alternative splicing, and it's controlled and regulated by epigenetic mechanisms and factors. But this understanding makes us ask a question: what is a gene? And maybe I'll be back to that question later, because today researchers are not sure is the gene at the DNA side or on the RNA side, or both sides. The situation is living right now. Okay, now we have to open this picture of alternative splicing. This image illustrates how alternative splicing works. Now, focus your attention on these colored segments, blue, green, red, light blue, and orange. They are called exons, and the cell's mechanisms have selected them from different locations. No, there is no one singular unit of information. Cell is able to pick these sequences, these exons, from different locations, even from different chromosomes. These sequences are joined together and transcribed into RNA. We are calling it precursor messenger RNA, pre-mRNA. The cell can modify this so-called pre-mRNA in different ways by combining useful exons and removing unnecessary introns. In this way, the cell can create various combinations from the transcribed sequences and further produce different proteins. And the most important thing is that the DNA has not to be modified. There's no need to change DNA at all. DNA stays the same. No modifications. This mechanism is epigenetically directed and regulated. And there are three main forms associated with this mechanism, and they are DNA methylation, RNA molecules, and histone epigenetic markers. A few examples of alternative splicing and its complexity. This is the most significant mechanism behind protein diversity, and again, of course, behind the biodiversity. And an excellent example of the power of alternative splicing, one gene called the SCAM gene, it's very common within insects. It can encode more than 38,000 different protein isoforms by using alternative splicing. And remember, DNA does not change. More than 38,000 different proteins. Put this information in the context of adaptation and variation. Now we understand why insects have a great capabilities to adapt to changing environment. It's also important to know that not a single protein in a human body is produced without RNA splicing or alternative RNA splicing. That means that every protein in our body is produced at least by RNA splicing. What's the difference between RNA splicing and alternative splicing is that if the cell doesn't use variation between these, if it doesn't use combinations with the exons, then it's only RNA splicing. But at least when there are combinations with the exons, then we are talking about alternative splicing. And we should conclude that there are no single information units called genes in the genome. And what's the most important conclusion is that DNA actually is kind of passive data repository. The cell can pick useful DNA sections, organize them, prioritize them, maintaining DNA integrity, and so on. And I'm not the only researcher who has discovered or understood that DNA is passive information. You know Dennis Noble? He said that the modern synthesis has got causality in biology wrong. DNA on its own does absolutely nothing until activated by the rest of the system. DNA is not the cause in an active sense. I think it's better described as a passive database, which is used by the organism to enable it to make the proteins that it requires. And the other studies have said that it's difficult to imagine how genes, composed of DNA, a passive molecule, can generate patterns in space and time. And although DNA transmits genetic information through time, it basically has a passive role. This is an interesting detail. Did you know that after a loss of information, meaning harmful mutations, deletions, and so on, the cell is able to compensate for the loss of information by programming the alternative splicing machinery to function in a more efficient and complex way. It's an incredible mechanism. So the programming, it happens all the time, but it's based on a code built in the cell. Here's a few nice-looking and funny examples of alternative splicing and its power. And cichlids, the adaptive radiation is basically based on alternative splicing. No changes in DNA are needed. Okay, let's get a little deeper. A few words of DNA compression. DNA is called around spools called nucleosomes. I think this was shown yesterday at most slides. We can see those protein structures called nucleosomes and histones. The spools are arranged side by side in groups, and the groups are placed sequentially and side by side. And this means we have a multi-layered packaging method. The packaging ratio is about 10,000 to 1, meaning it's incredible. That's why when we open up the whole structure, it's about 1.8 meters long. And the cell is able to open or close the structure by using chemical modifications such as methylation and so on. If DNA needs to be read, the structure is opened. This is the basics of epigenetics. If we unravel a chromosome located in the nucleus, we can observe that it is packed into what is called chromatin or chromatin fiber. Chromatin is made up of disc-shaped protein complexes called nucleosomes, around which DNA is coiled. Nucleosome contains sector-shaped structures known as histones. Each nucleosome contains 8 histones, and attached to each histone is what is called a histone tail. They're like lines or threads. These tails carry epigenetic markers, which play a crucial role in determining how and where the cell's mechanisms can read DNA. and especially how the final product, such as an RNA molecule or a protein, is constructed. And through histone markers, the cell maintains memory and the organization of information. It acts as kind of biological registry database for the cell. So it's like an operating system, and this is the registry database. I think it's a little bit more redundant and reliable than Windows 11 registry database. I hope so. Epigenetic markers can attach to the tails of histone with at least 20 different types known today. And researchers found annually from 3 to 5 more of these markers, new markers, because they are so small. The size is at atom level, so very difficult to observe them. And they have numerous control and regulatory functions. They're with the alternative splicing regulation. They affect the location of method groups on top of DNA. They influence post-transcriptional modification and post-translational modifications. And really, they function like a biological memory registry database. They synchronize and coordinate multiple layers of information to work together and in a timely manner. You may have heard the terms of 3D genome or 4D genome, spatio-temporal genome. Then we are coming to histone epigenetic regulation. Here's a wider picture of a histone and its tails, and you can see those markers on each tail. Those small parts, H3 and so on, those are those histones. And those tiny, almost atom-sized little markers are called histone epigenetic markers. And together they form an extremely complex code called the histone code. Scientists have solved about 1% of this code because it's so super complex. Over 20 different marker types, every one of them having a regulatory function. Their location on the tail, these all together make an astronomical number of different combinations. For some reason, I lost the mouse cursor, but let's see if it works.
Maybe draw it to the right. Right up there. Because on the screen.
Okay. Thank you. Yeah. Thanks for your assistance. So, because of the astronomical number of different combinations, the histone code is extremely hard to solve. So we have a long list of regulatory tasks those histone markers are involved in. They shape chromatin structure, they affect post-translational modification, alternative splicing, they maintain DNA integrity, they are interaction with other forms of epigenetic information, so they're keeping the lines with those hands. They regulate transposons. They prioritize which DNA sequences hold most importance when the genome is reorganized and rearranged. For example, the most important thing is to maintain sequences needed for DNA repair. And the second sequence is used by the immune system. Third, sequences needed for the production of key enzymes. 4, sequences related to metabolism, etc. And today, at least 20 different marker types. I have to make a correction. That was 22 is the correct number today. 22 different marker types. And it's important to know that combinations of these markers affect regulation on several cellular mechanisms. And the code is extremely complex. And this is heritable information. The cell uses non-coding RNA molecules as information transmitters. So we have epigenetic memory with this system. Then the RNA signaling molecules was the third form of epigenetic information. Did you know that there are more than 150 different epigenetic marker types known to attach to RNA molecules? On DNA, we know perhaps 4 or 5 different marker types. So this should help us understand that RNA molecules are the active agents carrying the most significant biological information. And how does the cell use RNA molecules? The direction and transfer chain of biological information in the adaptation of organisms, it can be described as follows. There are environmental factors, and then the organism's senses and receptors monitor these environmental factors. Receptors or the brain transmit information to cells through signaling pathways using various signaling molecules. And the cell's epigenome responds to the signals. The cell's epigenetic program possibly changes, and the function of the cell, tissues, and organs adapt to changes in the environment. And these changes are inherited by the offspring. And we're talking about mechanisms, no random mutation or selection. Long non-coding RNA molecules are extremely important. They have several important tasks. They regulate and control cell differentiation programming. They direct and regulate the formation of healthy tissue type. They mediate and control organs to work correctly. And they regulate and control body plan. Did you know that there are more than 9 times more of these complex long non-coding RNA molecules in our bodies compared to a chimp? This means that at the true information unit level, the similarity between humans and chimps is about 10%. Long non-coding RNAs, they don't tolerate mutations, meaning errors and mistakes. Even a small mistake can lead to serious diseases, such as cancer. And here are the most studied signaling pathways. I will not talk about this, not so much today, but I think most of you have at least heard about this. But they are very important in the epigenetic interaction and coordination, especially between cell-to-cell communication and, of course, environmental communication between the cell and the environment. We already know that epigenetic information is dynamic and it's reversible. It enables organisms to adapt quickly and efficiently to changing environments. It doesn't take millions of years. It takes possibly only a few generations or Sometimes it takes only a couple of hours. Organisms have various recognition mechanisms, we call them receptors, that allow them to gather information about the environment and living conditions. These are diet, for example, climate, sensory stimuli, pheromones, stress, toxicants, and so on. And cells use signaling pathways and specific RNA molecules and proteins to transmit this information. Rapid response is possible due to an efficiently functioning information layer called epigenome. And this is based on epigenetic readers, writers, and erasers. Wow. And this information is heritable. It can be inherited. And epigenetic readers, writers, and erasers make it possible to have dynamic and reversible modifications. And there are specific enzymes responsible for this complex information handling system. As we have learned, epigenetic modifications often function like switches. But to be accurate, they are not on-off switches, but rather dimmer switches, like turning a knob. Let's have a closer look. I could use the network because we have an interesting demonstration of this mechanism. It's loading. Oh, sorry. Maybe not. I'm not taking the time to use that, but let's go on. Okay, I will continue. There are different types of epigenetic memory in cells, and we can distinguish between them on how stable the epigenetic modifications are in terms of inheritance. So we know that epigenetic information functions like a memory for future generations. And interesting is that it also applies to harmful things such as addictions. And Darwin, in his theory, survival of the fittest, actually it's based on epigenetic memory, which is affected by temperature, food quality, sense of smell and taste, pheromones, stress, and so on. And this memory, especially by using histone epigenetic markers, it is passed to the offspring in the physiology of reproduction. And most organisms, they use extracellular vesicles for that mechanism. And in addition to the inheritance of DNA, non-coding RNA molecules transmit epigenetic information to the tails of histones in embryonic cells, which direct the setting of epigenetic profiles in the embryo and fetus. Methylation profiles, DNA methylation, they are usually weakly inherited. Maybe to the next generation, but not so far away. Oops. But we should know that there is a much stronger and stable mechanism to transmit epigenetic information across and over generations. This means traits and characteristics, adaptational states, etc., they are stored in the histone database. And then non-coding RNA molecules encapsulated in the extracellular vesicles transmit this kind of complex information for the next generation in the physiology of reproduction. It's a very complex mechanism. Okay, a very recent discovery is the mechanism by which a cell can deliberately modify both RNA and DNA sequences. So by using specific enzymes such as ADAR or ApoBec enzymes, the cell can make intentional changes in DNA-based sequences if necessary. And again, this emphasizes the passive role of DNA and makes the mutation theory to be even weaker. You can find more information by using keywords "mutations are not random." As we already learned, alternative splicing is the most significant mechanism behind protein diversity and biodiversity. Most of so-called speciation events are based on alternative splicing. The second mechanism resulting in speciation is DNA rearrangement after harmful mutations, or a loss of or corruption of information, or chromosomal damage, for example. But there are less studied mechanisms that have been discovered recently. RNA editing is one of the most fascinating mechanisms by which cells can modify different RNA molecules at the RNA base level. And in this way affect protein structures or their functionalities. So we know that a cell can change any RNA base to another in a controlled manner. The most studied one is RNA A to I editing. And this, of course, affects organismal change and maybe leads to so-called speciation, meaning that speciation can occur without a single change in DNA sequence. But there are side effects of epigenetic regulation because it easily causes errors in DNA. And that leads to observation that cell rearranges DNA. And this often leads to variations in the organism's outlook. And this is an important thing to know. You can read it by yourself. So it's an incredible system because human engineers are using similar technology in the network address translation called NAT. It's a masquerading technology. Yes, it means that as we can read, HR refers to a modification of adenosine into inosine. Inosine is not a base typically found in RNA. Rather, adenosine has been modified by an enzyme from the ADAR family, transforming it into inosine. And when mRNA containing inosine goes through the translation process during protein production, the translator, meaning ribosome, interprets inosine as guanine. So it looks like guanine, but it's inosine. Yes.
Sorry.
Yeah, it's okay. Okay, we are familiar with these verses. I passed them very quickly because of the tight schedule. We all know the noun min is used, and we know that in the creation account it refers to a species, type, group, class, category, breed, etc. And on the King James translation, if it's translated as after his kind or after their kind, we know that already. And in taxonomy, kind corresponds to a family, sometimes a genus, as we learned yesterday. But this is important to remember. The phrase baramin, meaning after his kind, this is not used in connection with the creation of man, meaning that God, and also written that God created man in his own image. In the image of God created he him, and today especially, male and female created he them. And did you know that there are specific mechanisms maintaining the number of chromosomes in the human cells? These mechanisms are so-called NORAD, long non-coding RNA molecules, together with the so-called pumilioproteins. They maintain our chromosome number to be stable. That's why we are not experiencing any kind of speciation, despite of the fact that there are 1.9 million genetic defects in the human genome worldwide. Almost 2 million. Epigenetic regulation is associated with hotspot mutations. And here's the mechanism, how it happens. Methylation of DNA, it predisposes the genome to errors, to mistakes. And the most common methylation-related genetic error is the conversion of methylated cytosine to thymine in a process known as deamination, which is caused mostly by oxidative stress. And if a base in DNA changes, then the DNA repair mechanisms attempt to restore DNA integrity by correcting those errors. However, the cell doesn't repair the conversion of methylated cytosine to thymine. And if the cell is replicated, meaning copied, the error becomes permanent and is usually inherited, which means that errors accumulate from generations to generations. And this results in the fact that so-called GC content gradually shifts to AT content. And there are side effects, negative consequences. It means that there's a loss of genetic diversity. The cell attempts to rearrange DNA. The cell's ability to produce a variety of proteins weakens, and the immune system weakens, and eventually the organism is under the threat of extinction. And we already learned about those CPG sites and CPG islands. CPG site means that cytosine can become methylated. At CPG sites, cytosine can be methylated easily at CH3 groups. And CPG sites are sometimes rich in specific regions, forming CPG islands, such like this, meaning there are a lot of CG pairs side by side. And the cell uses these CPG islands for different purposes, such as promoters and enhancers. and initiations, transcription start sites, and so on. So they're very important markers for transcription machineries. And when cytosines in these CPG islands gradually convert to thymines, regulatory regions stop functioning. And there are consequences. Production of crucial protein stops, organisms' traits and adaptive capabilities weaken. And a good example is the flightless birds. It has been investigated that they have lost a lot of CPG islands, those extremely regulatory regions. And this is the fact that destroys the entire theory of evolution. The cell has no mechanism to rebuild new CPG islands. There is no mechanism for that. And this means that genetic degeneration, genetic entropy, it is inevitable. However, the cell tries to balance GC80 contents. Because low GC content, it has negative side effects. It makes it more difficult for the cell to construct functional and important RNA molecules. It causes chromosomal damage. Balancing occurs during reproduction, specifically in meiotic recombination. And when DNA is rearranged, then we can observe a rise of activity within transposable elements. It's also associated with joining of broken chromosomes and chromosome fusions. And it leads to a decrease in the total number of chromosomes. And there's rapid genetic degeneration due to this phenomenon. For example, the human genome contains almost 2 million genetic errors worldwide. You can check it from this GNet site. However, as we learned, the human chromosome number is not reduced. Humans don't speciate. Mutational load is a biological fact. And a few years ago, the leading evolutionary biologist claimed that more than 95% of the genome is useless junk. You know, the junk DNA theory. And they thought that the junk DNA works as a buffer against harmful mutations. For example, Beetz and Myers say that if much more than 3% of the genome is functional, evolution becomes primarily a destructive process. But today we know that there's no junk DNA. There are studies concluding that there has been an explosion of research addressing possible functional roles for the other 98% of the human genome that doesn't encode proteins. In fact, more than 90% of the human genome is likely to be transcribed. So we know that mutational load remains, but there's no buffering junk DNA anymore. What are your conclusions? That's why we are talking about information loss and corruption of information. And we know that it has a lot to do with so-called speciation. We know that epigenetic regulation results in genetic errors, and this can easily be observed as a rapid chromosome loss, especially within diploid organisms. Can it provide us an excellent example? Remember that after a loss of information, the cell rearranges the genome by using prioritization principles. That's why there are no messy outcomes. such as messy wolves or messy dogs. We can only observe functional and good-looking phenotypes. But we can observe a rapid reduction in chromosome numbers. And we have to understand that this has a lot to do with epigenetic regulation after adaptational events. And I have more examples of this phenomenon. Horses, muntjacs, mice, Portuguese ancestor 40 chromosome, and after only 500 years, 22 to 30 chromosomes. Only after 500 years. Giraffe. So, Lenski's experiment provides us an excellent example of a genetic entropy. It started in 1988 using E. coli bacteria, but this has been a disaster for the theory of evolution. Today, after nearly 90,000 generations of bacteria, evolution has not occurred, but the opposite. And almost 3% of bacteria's genome has already been lost. They are threatened with extinction in the next 15 to 20 years. The only significant adaptive event, citrate utilization, was based on DNA rearrangement, no novel information. Did you know that deletions, how do you pronounce that? Deletions are 4 times more common than any types of insertions. And yesterday, Andy had a very good question regarding transposable elements and those insertions. And we should know that deletions are 4 times more common than any insertions. That's why the genome is going down. Okay, it's time to make conclusions. We have observed epigenetic regulation or loss of information, no evolution. And we can conclude that Darwin was wrong, absolutely wrong. We know that diet is the most significant factor influencing epigenetic regulation. Random mutations were not the cause of the finches' adaptation. And we know that natural selection is an imaginary force. Changes occur rapidly, just in a couple or a few generations. And we know that any change in organisms is based on pre-existing biological information or a loss of information that results in a rearrangement of information. The genome of finches inevitably degenerates. You can explore the topic using the search terms Darwin's finches, diet, epigenetic. More conclusions. Adaptation of organisms leads to observable variation within species types. And from the Bible, Baramin, it means after his kind, not before his kind. Dogs remain dogs, bacteria remain bacteria, as is written in the Bible. Variation can happen very rapidly, even within a couple of generations. And it is based on epigenetic regulation of existing biological information or the loss of information. All the information necessary for adaptation is already present in organisms. We know that there is no mechanism for evolution. Organisms don't acquire new biological information that would lead to increased structural or functional complexity. And based on these observations, all organisms are genetically deteriorating. We can observe a rapid decline in chromosome numbers. Today, there are nearly 2 million genetic mistakes aroused in the human genome worldwide at the population level. However, this has not led to human speciation. The number of completely, fully beneficial mutations is 0 in the human genome. No beneficial mutations at all. So we can ask, why would one of the most important questions, the questions of the origin of life and humanity, be written metaphorically? Because based on scientific observations, the biblical creation account is based on true events. Creation is mentioned 243 times in the Bible, but evolution as a word, never. The truth can be found in the Bible. Thank you, and God bless you all.
So thanks a lot, Tomi, for this thorough and mind-blowing exposition of epigenetics. So pointing out that we really have to abandon, as you mentioned, the classical dogma of biology that one gene codes for one single protein through messenger RNA. So, and that this really sort of destroys Darwinian evolution. So now we have time for questions. So, Antti Rettingfors.
Thank you, Tomi, for an amazing mind-blowing talk. That was incredible. I've just got a couple of questions. One of the implications would seem to me from epigenetics that if we live well and if we believe, and basically I could even apply between a prayerful spiritual walk, but certainly if we live well, just talking about physical living, we should be able to pass on that, some aspects of that, to the next generation, which is mind-blowing again.
I'm not sure about the line. Is it open? Sorry, this technology microphone.
Nosta sitä ylemmäs, se on siellä.
Yeah, okay. So the question was that if we...
So my question is that true, or am I on the wrong line?
Yeah.
What is true? If you live well.
If we live well, so does the information be passed to the next generation? Yes, absolutely. Because the epigenetic mechanism and factors, they really are capable of transmitting this kind of information to the next generations. That's incredible, yes. Meaning all of our attitudes, all of our what we have done in our life, our physical activities, all kind of that. But also our brain attitudes and so on. Maybe not what we believe in, but at least our attitudes, mental things like that, yes.
And the second very quick question is this published in, you mentioned a book, I haven't seen it up on the screen, your book.
Yeah, most of this that was just presented is published in my book, and we are planning to translate in English in the next few.
I'm so interested.
Oh, it's good to know. I will.
Sorry to go.
Yeah, okay. Yeah. so far in Finnish, but after a couple of months, we hope in English, yes. Yeah.
Thank you, Tomi.
Yes. Okay.
So you mentioned that the BCAN gene produce 38,000 disease proteins.
Yes.
And so my question is, how redundant are these proteins? Or do they alter the disease function?
Yeah, that number is a theoretical numbers of possible combinations. So I'm not sure about the living nature, how many proteins insects have produced, but it's a theoretical maximum of combinations. But of course, there might be problems with the redundancy of those proteins. But as we learned, there is a compensatory mechanism. The cell can compensate for the loss of information and for the mistakes the cell makes. Yes. Sorry.
Thank you for a really informative and inspirational message. This is kind of a little bit different question, but the information in the DNA proves there is something of intelligence behind it, but the information in itself is of course not intelligence. It's kind of a static thing, as you said yourself. The DNA is static in a way.
Yeah.
But everything that reads this information seems to be the intelligence or the intelligent thing agent acting upon the information here. From an epigenetic standpoint, where do you see the intelligence acting here? Or could you almost say that the intelligence needs to be even outside of this whole structure to be able to actually read this information the way that epigenetics suggests it does?
Do you mean, is there built-in information?
Well, in a way, I kind of see, to put some more fruit into the question, it looks a lot to me like how the Bible is made. Because the Bible is information, it comes from somewhere else, it's static in a way, it will never change, God has promised that. But to be able to read the Bible, you need to piece together things from kind of like an alternative splicing in a way. You know?
Yeah, yes.
To get truth.
Yes.
Yeah, but it needs to be, at least when it comes to the Bible, it needs to be revealed from an intelligence outside of the agent reading the information. And epigenetics to me suggests it's exactly the same thing happening actually throughout the whole world.
Yeah.
And you need an intelligence outside acting upon the agent reading the information to make it meaningful. In a way.
Yeah, that's a good learn. You are a learner. You are a teacher. Thank you.
Well, would you agree? It looks like that.
Yeah. I agree with that. Pretty much, yes.
Okay. Well, sorry, that was a good question.
Yes, interesting talk. I was thinking, could you make the hypothesis that all the kinds of animals have more chromosome counts before the flood? Like every today they had much more chromosomes than we have today, perhaps.
Can you repeat the question?
Yes, because I was thinking of the decline in the chromosome counts.
Yes.
If one can make the hypothesis that before the flood, all animal kinds had higher amounts of chromosomes, because we have 23 today in our human cells, could we ever have it?
Oliko ennen vedenpaisumusta enemmän kromosomeja?
Before the flood. Yes, absolutely. And we can follow, we can make a line and count and estimate the number of chromosomes by going back a few thousand years and so on. And we should also remember that Noah was told to take bara means on the ark. So they were the basic types of those kinds. Yes.
Thank you. I'm thinking about heritage here, and you mentioned that changes occur in fetuses and embryos, but does it also occur in sperm, or is it actually females determining the heritage level of epigenetics?
Both females and males transmit epigenetic information by using extracellular vesicles. So there are those nano-sized bubbles, grease bubbles, in the sperm fluid, in our men's sperm. And female system is a little bit different, of course, but both of them are carrying epigenetic information by using mostly long non-coding RNA molecules in those vesicles. It's an incredible mechanism.
About the original information, here we have over 200 different codes used in biology, and it's noteworthy that the information is encrypted. It's sent and it has to be decoded.
Yes.
And this type of information comes absolutely with 100% security from an intelligent source outside of the Earth.
Yeah.
We have time for a last question.
Well, first of all, thank you very much for this beautiful overview of all these epigenetic mechanisms. I have actually a question. You said, at least this is what I understand, that we see in fact the number of chromosomes decline over time, and this would be. Is it always a decline? Do we never see fissions or fusions? I mean?
As I mentioned in the presentation, it applies mostly with diploid organisms. But we know there are polyploid organisms, and then we can observe, in some cases, increasing chromosome numbers. Or they sometimes talk about duplication, whole genome duplications, or part duplications. But they are based on epigenetic regulation too.
Okay, and is it so that if I understand correctly, that you say that this is sort of rescue mechanism?
Both, yes. Both of rescue, and it's also associated with the adaptation needs, yes.
Is it not a spontaneous mechanism that occurs? Or
Sorry?
is it not a spontaneous mechanism that occurs driven by?
It's spontaneous. It's triggered by environmental signals.
I have a feeling that appreciation is an intrinsic property of genome, that it is always making novel phenotypes without. I think it could be so because of a cue from the environment. This could be a genetic driver. But I have the feeling that God created something about him with an intrinsic propensity to form novel species, without using novel information, because all the information is already there.
Yeah. Okay.
Frank, you had the last
Yeah, haluatko pitää pienen? Just a second. Oh, sorry.
Just, at the time for 20, 25, 30 years ago, I studied a lot of epigenetics.
Yeah.
And then we went away from epigenetics in my research. And I don't remember fully why. But it's very impressive to listen to you. Marvelous, I've learned a lot. And I'm so impressed that you have
catch up with all these things in 2017. That's marvelous. I'll just see that the focus here on kinds of may also lead us to a little bit kind of simplifying. I think the complexity of what God has done in, the transcription factors, the function of the RNA in itself, the functions of the proteins in itself, which is then directly from the DNA. So you say one DNA, one RNA, one protein, that's okay. But it's still the main codon is there, DNA, RNA,
no.
protein.
No.
Yes.
No. It's not there.
In my service, in my research, is there. But as you said, I mean, when you talk about all this, what we call LNC RNA, in itself is transcribed from the DNA.
Mm.
So it still has its own information in itself, even though there is epigenetic changes.
Do you know that most of non-coding RNAs are products of alternative splicing machinery? There are no direct transcription of the certain DNA sequences. They are constructed and built in similar way as RNAs used for protein production.
I think what you mean is that the codes of these RNAs are already present in your DNA.
No, it's not.
So
there are no single genes producing RNA molecules. There are no single genes.
About genes, this is the point.
This
is the point. The whole concept of genes has changed over
yes.
the last 5 years. This is what I understand also. But the thing is, if you make RNA from somewhere, you must transcribe it from your DNA.
Of course.
So it is the code.
Of course. I'm not saying that DNA is not unnecessary. Of course, it's extremely well organized information. It's incredibly organized information, but it has no dictative role. It doesn't determine our traits or characteristics.
I agree. People used to say that the cell is in control. It's a common theme these days.
Yes, epigenetic mechanisms control DNA at all levels, at every level.
You say that the cell is in control.
No, epigenetic mechanisms control. Yes.
No, it is a holistic system.
Yeah.
And alternative splicing is explained how it works in our new book with Tomi Aalto. Game over, Darren.
Okay. Thanks a lot for your care. This is a signature of an excellent talk that initiates many questions, and I'm sure we can continue now the discussion during the coffee break.